we know that animals have amino acid requirements since the 1930s when we discovered the amino acids, and they started doing feeding trials in rats and mice, and discovered a lot of information about them at the time. One of the challenges over time, has been that there's too many of them. Okay, it's not like one thing, so we could easily optimize this.
Yep.
And we have at least 10 that we have to pay attention to in dairy, probably, maybe a few more, because even when we've had some looks at some of the non-essentials, the effects are small, but they're significant.
Welcome back to the Dairy Podcast Show, brought to you by Wisenetics. My name is Barry Bradford from Michigan State University, and today I'm very pleased to welcome Dr. Mark Hanigan from Virginia Tech to the show. Dr. Hanigan, thanks for joining us.
Yeah, happy to do that.
So I've watched your seen your research for many years, known you for a while, and one thing we have in common is we're both a couple of idiots out wandering around, right? So you grew up in Iowa. Can you talk me through your path to get you to where you are now?
Yeah, well, it's not a straight shot. Okay, so I tend to do things backwards for some reason. I sort of like the analogy. I think I've forgotten the guy from Oklahoma that in the 1930s that always did sort of old home cliches, but he said there's three kinds of men in the world, or people. Okay, men in the 30s, but people are. This is really a men joke, so my apologies. But he says there's those that can learn just by reading, and then there's another set, and the first set's a very small set. Those that can learn by watching others do, or listening or watching others do things, and he says, then there's a whole bunch of people that just got to pee on the electric fence to find out what's going on. But I grew up on a farm, a multi-species farm in Western Iowa, and I decided I was going to make my fortune farming. And so I restarted the dairy. There had been a dairy on the farm for a short period of time, but it had been closed down. And so I restarted that, and that was in the late 70s, when interest rates were about 12%, and then they went up to 20%, and then they started knocking down price supports. And, timing is everything. And I probably wasn't the best dairy farmer anyway, because I didn't have a lot of training in it. And there was just too many cards stacked against me. So I exited that business and decided, well, maybe I should go to school after all. So I went to Iowa State, got a degree in dairy science. And while I was there, I had some really good advice from my wife's uncle, who was research director for swine for Wayne Feeds. He says, with your background, you really should consider getting into like professional consulting or, get a PhD. He says, you're going to have a lot of value because you have the field experience as well. So that sort of turned me on, and I did that. So I went to UC Davis, got a master's in animal science and a PhD in nutrition, stayed on and did a postdoc in biochemistry for a couple of years. It was another downturn in the economy, so there was, I think, 3 jobs in dairy science. That's where you look for jobs at that time, right? And the whole year, only one of them was a nutrition job. I applied for and interviewed at Idaho, didn't get the job. And so I ended up taking a job in dairy research at Purina Mills. And I worked there for 12 years doing, a mix of research and sort of what might be called extension or field support.
Yep.
And then Land O'Lakes had bought them, and they were contracting. And so I was, invited to maybe look for a job elsewhere, along with about 200 other people as they tried to close their budget gap. And so I had already interviewed at Virginia Tech by that time because I could see the writing on the wall, and I'd interviewed at several other places as well. And so I took the job at Virginia Tech, and that was 20 years ago. And so that's where I'm at now.
That's amazing. And I'll say this because you won't, but having a huge impact through your research and outreach at the university there. And, maybe it's a good lesson for some grad students or young people out there that sometimes things look like a really rough, negative thing. And maybe in the long run, it's the best, right?
Yeah. I mean, certainly I learned a lot from all those different steps,
Yeah.
And again, I had to pee on the fence everywhere. Okay. I couldn't be taught without that.
Adiseo, a global leader in nutritional solutions and the provider of Smart Amine M, the best in-class rumen-protected methionine product for dairy producers who want to optimize milk production, capture more value from their components, and maintain the lifetime performance of their herds. For more product information and to calculate your return on investment when you balance your feed with amino acids, go to milkpay.com.
Well, okay. A lot of your contributions have come in the space of protein nutrition for cattle in general, and especially dairy cattle. And you've been working a lot on. I would characterize it as sort of what's really the driving factors for getting animals to respond to additional protein or amino acids. Is that a fair characterization, or how would you frame what your main focus is?
No, I think that's fair. I mean, having been trained in sort of nutritional biochemistry and biochemistry, I tend to like to start, with the basic science side. I mean, yes, there's an overriding question at the animal level, right? And so Lee Baldwin was my mentor, and he was fairly adamant that you have to drill down to sort of understand. If you really want to understand the problem, you need to drill down and understand the underlying mechanisms, and then it's important to integrate back up to the whole animal level, and that's part of what he taught us there, and I still ascribe to that. I think that's the way to go.
So I think most people, with some dairy industry experience listening to this, if you think about meeting protein amino acid needs of cattle, a lot of people are right away going to think about bypass amino acids. So I wanted to talk with you a little bit about that today. So how have you seen that evolve in the last 20 years, I guess, first of all?
Well, I mean, we know that animals have amino acid requirements since the 1930s when we discovered the amino acids, and they started doing feeding trials in rats and mice, and discovered a lot of information about them at the time. One of the challenges over time, has been that there's too many of them. Okay, it's not like one thing, so we could easily optimize this.
Yep.
And we have at least 10 that we have to pay attention to in dairy, probably, maybe a few more, because even when we've had some looks at some of the non-essentials, the effects are small, but they're significant. Okay, so they're also playing a small, minor role, and I don't think we're going to get in too much trouble ignoring them, unless we can somehow figure out how to design diets, with cost-effective feeds that have no non-essentials in them. That probably is not going to work. So, I think that over time, then, we've looked at the mechanisms, and one of the key things that we discovered in the 90s, while I was still at Purina working on a project like this, was that there wasn't really a single limiting amino acid. I mean, when we tried to use those models, okay, and I've always because of Lee, mixed modeling together with science, or I shouldn't say with science, because modeling is an arm of science.
Yeah.
Mixed it together with experimental research. And when we gathered a bunch of data where we'd infused different groups or individual amino acids in cows in England and Scotland, the model explained none of the variation that was there, because it was basically that says it's the wrong model. And it was this first single limiting nutrient idea. And so the only way we could get it to work was to consider multiple amino acids. And then when we looked at, okay, well, what's going on with the variation that's there in the animals in reality that we're not explaining with the model? Well, then another big chunk of it went with energy. So it makes sense. You got to have energy, and you got to have multiple of these essential amino acids, and probably enough nitrogen to make sure everything works. And so you can't really, not consider what's going on in the rumen, but it's also an important part of this. But I think the transition to your question has been the understanding that we can't just look at protein bypassing the rumen and think that we can optimize everything based on that. We can, right? If we just flood it with protein, which is what we did for years, then we can get everything good, okay? But the efficiency is not good, and that's leading to all these nitrogen problems in the groundwater and air emissions of ammonia, et cetera. And so I don't think society, both from an environmental and health standpoint, nor from an economic standpoint, allows us to do that anymore.
I think you're probably the one that helped me eventually decide that I needed to chop up my rain barrel. So many people at some point in their education, have been presented with this nice talking point to understand what a first limiting amino acid means. Can you explain that if people don't remember hearing that before?
Yeah, it's an interesting story. I mean, I sort of like the history part of things as well. And
Yeah.
if you go back to, it was credited to von Liebig, who was, oh God, I don't remember if I'm going to have my dates correct, but it was, I think, the early 1800s, like the 1830s. He was a German plant scientist, and he'd observed, or he's postulated, I guess he was in the 1870s, he'd postulated that, if one nutrient was limiting in plants, then the plant can't respond to any other nutrient. And he was looking at nitrogen and phosphorus. When you dig back farther, Sprengel actually was ahead of him by about 40 years, and he'd observed that already. So von Liebig was a better promoter, I think, and he
Okay.
so maybe he had podcasts or something, I don't know. But he gets credited with all this when in fact it was Sprengel that actually observed this. But Sprengel wasn't so forthright in his statement. He just said, when a nutrient is less than the plant desires, growth will be proportional to the addition of that nutrient. He didn't say that they can't respond to any other
Okay.
nutrient. And so the fact that all this was, written in German, sort of precludes me from digging in, really deeply. And of course, it's hard to get a hold of some of these old documents. So I don't know if von Liebig had some other data that showed that it really couldn't respond to other nutrients, but it was that then that got used by Bloch in the 1930s when we discovered all these amino acids. Like, okay, how can we make sense of these? Right? We got 20 of these things. Okay, we have to figure out some way to conceptualize these, and so they conceptualized this idea of a old wooden barrel, that we used to use for catching rain off the eaves. Or I remember my grandpa storing his oats and things, that he was doling out in little scoops to the dairy cows. He had the 10 dairy cows in the barn. And if one of the staves is broken off short, then when you try to fill it with water, it's going to run over on that stave. And so the idea is, okay, well, that is the limiting nutrient. And that is the one that we have to add. And of course, from von Liebig, we're going to say, well, there's no other nutrient will help. It won't work. So it's a simple concept. It was easy to understand. It's in every darn nutrition book you can find. I wish I could come up with something that had such a large, wide impact, because even in China, South America, everywhere you go, they all know that analogy. Okay. I don't think there's any way I'll ever.
Even though now you have to explain that, to new students what a stave is, what a barrel is, right?
Exactly. We need to find a new picture of something, okay, that's no longer in production unless you go to a whiskey factory. So.
And so I think you kind of alluded to this, but just to make sure, we're making it clear, that model says that if the one amino acid that's the furthest from the requirement is the only one that should benefit animals' productivity or growth or whatever. And how does your data sort of refute that or not fit with that?
So, I mean, like I said, we showed that at the animal level with infusions that this didn't seem to work very good conceptually. So then we dug in after I got to Virginia Tech and had more latitude to work on, sort of basic things and looked from a cell culture standpoint. Okay, well, how do these cells, these mammary cells that we isolated from lactating cows, how do they respond to these individual amino acids? And in fact, if you go back and look, there's a J.H. Clark at Virginia Tech, or Virginia Polytechnic at the time, in the 1980s, observed this, that if you took cell explants at that time and he made them deficient, and added an amino acid, he added methionine and got a response in total milk protein output. Then he took away the methionine and he added, I forgot what, histidine, arginine was one of them, I remember that. But he did all, I think he did 6 or 7 or even maybe 8 of the essentials. And he got responses to 3 or 4 of them on the same base media. Well, that is completely throws that barrel and stave idea out of the water. You can't have a response to multiple ones from the same base. And so we did a lot of work on that. And what we found really in the end is that, yes, we can't make more milk protein or more methionine in milk protein than we have methionine coming in. But remember, we're only converting about 30%. So it's not like we really have a deficiency.
Yep.
And it turns out that the regulation of protein synthesis, because it's a very expensive process from an energy standpoint, is really the primary regulator of all this stuff. And it responds to multiple of these amino acids and apparently some of these non-essentials as well. So it was the regulation that was holding it back, not the the actual mass balance. And the regulation had plenty of room for flexibility because it can manipulate the transporters. It can pull more away from the other tissues. The liver is just getting rid of it if it's not being used. It doesn't really have to have all the nitrogen it's using. So there was plenty of stuff floating around.
It's a great explanation. So if you're listening and this is triggering questions in your mind, I encourage you to get online and engage. There's a great opportunity to throw some questions in and get feedback from Dr. Hanigan or others that are part of the conversation. So, Mark, let's shift a little bit to sort of, people that are out there trying to run a dairy. Thankfully, they're not paying 20% interest, but it's still not easy to make a living. So, put this in context of running an entire dairy. Like, should they be thinking about rumen-protected amino acids? Is it cost effective?
Well, I mean, and of course, I spent 12 years in industry where those were the primary questions. Okay, because if it wasn't cost effective, it wasn't going to be a product idea. So, I'm very much in tune with that. And the answer to that, Barry, it really depends. The answer today won't be the answer tomorrow, right? Because it's an input cost versus output value, right? And not only on the input, costs are not only associated with the input. Today they primarily are, but if you go to some areas of the country like Florida and some of the Chesapeake Bay region, we have restrictions. There's sort of harsh roadblocks, right, or fences, right, that say you can't go beyond this in animal numbers because of nitrogen and phosphorus balance problems on the farm and causing problems in the water systems. If we start to monetize that on more of a nitrogen excess and say, okay, instead of saying you can only have so many cows, let's say you have a license for 1,000 cows. Well, if we were to feed the cows more efficiently from a nitrogen standpoint, then you could perhaps have 1,500 cows. Okay?
Right.
So if we start to monetize excretion in grams of nitrogen per day rather than just counting cows, we set up a different cost structure for this, right? In that case, that nitrogen excretion has a cost. And so if we pay a penalty for an environmental thing, the answer becomes totally different. So when we've done some optimization with that, if we optimize and put a modest cost on nitrogen excretion, that at NASA 2021 model, using the amino acid equations that are in there, it'll try to go to like a 12 or 13% protein diet. And even with amino acids, these rumen-protected amino acids at 10,000, a kilo, it will still use the heck out of them, okay? If I don't put a cost on it, no, it doesn't. It goes to like 15, depending on the cost of dietary protein. And it might add a few of those in because it alleviates some space problems, right? I mean, if you got a high-producing cow, you're always worried about or trying to deal with lack of formulation space, right? You got to get energy, you got to get fiber, you got to get all these vitamins and minerals, you got to get these amino acids. If you alleviate some protein in there and let it use more starch or more fiber, and then top it back up with amino acids, that has value in the formula. So it's really hard to give an answer other than when we've done this, it almost always uses amino acids. So it says they are cost effective even at today's prices. Now, milk protein, I don't keep track of it on a daily basis. I know it's been lower, so maybe over the last 6 months, no. So I think you have to say, okay, we're gonna let the optimizer decide what we're gonna do, right? We're gonna give it these ingredients and we're gonna say, give us the best diet to meet this production, and let it go, and then trust it.
And just to make sure everybody's following, like the basic concept is you can give up some total amino acid flow to be absorbed to the cow if you're making sure she gets some of the most important, maybe most limiting. Even though we're throwing out the rain barrel, there are still some amino acids that per gram are more effective at driving milk protein than others, right? So we can feed less total crude protein, backfill with a few targeted amino acids, and get to the same endpoint probably. Is that a good way to say it? Yeah.
That's correct. And what it turns out is, that there's different slopes on those, and we can talk about slopes, but it basically is the relationship between the metabolized amino acid coming in and the amount of milk protein we can get out. And methionine and histidine, have roughly about 2 grams of milk protein per gram of metabolized amino acid coming in. Threonine is not even in our model right now, but we've just finished a study that's being defended next week. And she showed that we also got a response to threonine. The isoleucine gets about 1. So, I mean, you have to look at the cost of the amino acid, right? I mean, if it's a 2 to 1, what's the ratio? What's the value in the formula? I really can't answer it, very well,
Yeah.
other than saying the diets we're formulating want to use these things.
That's fair. And you also brought up something else I wanted to talk about. You said, simplifying what you said a little bit, basically methionine and histidine both have about an equal efficacy to improve productivity. We've had methionine products available, bypass methionine available for many years. We've had lysine more recently. It's widely available now. Why not histidine? Why are we not feeding bypass histidine?
It's an economic question at the moment. There's couple of companies that have histidine products ready to go.
Okay.
Milk prices have been low, and so it's not been a good environment to launch those in. The efficacy of those products is actually pretty good, but the swine people and poultry people don't use much histidine, so the cost of producing the raw histidine is relatively high. So it's really the chicken and the egg problem, right? I mean,
or the bacon and the egg.
We could get more volume if histidine was cheaper. Okay, but I think the return is actually already there. They're just scared that the price is high. It's going to be a sticker shock to people because it's going to be higher, I think, in general than methionine is.
Interesting.
Asepsis, an ounce of prevention. Asepsis LLC is a Wisconsin-based animal wellness company focused on the development of state-of-the-art animal hygiene technologies. Our company's mission is to apply innovative hygiene solutions into animal agricultural and veterinary applications, improving the wellness of the animals with a focus on sustainability. We accomplish this through the continued enhancement of our chlorine dioxide-based technologies within a wide range of agricultural hygiene applications, from facility and animal hygiene to water treatment.
I think another thing that often comes up, at least with nutritionists, is, well, to what extent can we be smart about picking rumen bypass protein sources as opposed to individual amino acids and get to some of these same endpoints. And when you say you're optimizing a formula, obviously that's part of what you're talking about. But if somebody's not, used to doing this, how would you approach that in terms of ingredients to consider?
To start with, I mean, obviously the amino acids that are supplied by RUP are almost always going to be cheaper per gram of metabolized amino acid than they are by providing them with an rumen-protected amino acid. So we want to focus on providing, using up those resources first, which is good environmentally as well, because a lot of them are byproducts,
Yep.
and other species can't necessarily use all of these. So, we need to let the ruminant make use of those products the best we can. So yes, we should be using those byproducts first. We want to make sure we top up the RDP for the microbes first. We got a sort of a target of 10, but I think that's too high. I mean, we're operating above 100% below 100% efficiency when we do that. There's no reason we can't use a little less. Yes, it will reduce microbial protein a little bit and make us rely more on RUP, but I think environmentally that's a better story. So once we've figured out how much RDP and how much microbial, then we have to provide the rest as in the form of RUP. And again, those are cheaper sources, so we're going to use the ones we can. And then if we use an optimizer, it will tell us, okay? I mean, if we do it by hand, we're not going to know, right? If we're out, and we see this, right? We get these diets in from nutritionists. They're perfectly good diets. The cows are milking very well. There's nothing wrong with the diets in terms of animal performance or animal health. Mm-hmm. But when we optimize, we can save 30 cents a cow a day. Now, I know from my time in industry, okay, 5 cents a cow a day lost you the business, okay, on a large dairy, okay? 30 cents a cow a day is a huge thing. So it's not that you can't do it by hand or do it the old way, but you're going to get blown out of the water by younger people coming in using these optimizers, I think, eventually. And probably rightfully so, because you need to keep adopting new technology. So they will use these things and top them up to whatever the best solution is for your cost structure today, your goals today, your environmental cost, if there is any today. And I can't really tell you exactly what they should be. The targets that we put in the NASM, I think are reasonable targets. They may be high. They're efficiency targets. I think they may be a little high because they don't really consider lost production as well. Okay, it's just what diets provided the best efficiency. Well, those might have all been low production diets that we don't want. I don't know. I just rely on the optimizer to do it.
Gotcha. And that does bring up, I'll just point out for anybody listening that the nutrient requirements of dairy cattle, which is the Bible of dairy nutrition, Dr. Hanigan was a key member of the committee that brought out a new version of that about 3 years ago. And sorry if that causes PTSD, bringing up the many hours of work on that, but thank you for your service on that.
That's another example of me not being too smart. Okay, who would donate 7 years of their life to do something for free? Okay,
that does bring up one more fun question I wanted to ask you. At the risk of offending somebody out there, if you look back at all the different jobs you've done, you've been a full-time dairy farmer, you're a professor. Day to day, what was the most enjoyable job for you, do you think?
Well, I don't know. I mean, I always liked chopping cornstarch in the fall. It felt like you got quite a bit done. You got to drive around a lot, look at things. The weather was nice, etc. I don't know. I mean, I certainly have enjoyed science in general. I mean, it's a joy for me to work on these problems. Not every day. Okay? I mean, particularly bench science, when you're trying to, whenever we got to develop new techniques, I hate that because it's like this, you never know whether it's even going to work in the end. You don't know if it's going to take 1 year, 2 years, 5 years. You don't know how much it's going to cost. You don't know how much value it's going to provide in the end. So it's a lot of unknowns. But once you get one of those working, something new, and you can actually then answer some more questions, well, then it's a lot of fun. And maybe it's like childbirth. You forget all that pain that happened before.
Yeah. Or I was going to say, it's like harvesting corn silage, all that work all summer,
Yeah, I forgot about all the cultivating and all that stuff that they don't even do now. They just go around and spray everything all the time.
I know.
We had to do all this stuff.
It's time for our famous 3. Expand your feed possibilities with a trusted partner. You can count on Schooler for consistent, high-quality feed ingredients with reliable delivery. Access an extensive portfolio of product options that fit your unique rations and cost parameters and get the essential nutrition solutions for your herd at all stages of life. Visit schooler.com/feed to learn more about the benefits of working with Schooler. That's Schooler, SCOULAR.com/feed.
Oh, okay. Well, we've got 3 questions we've got to ask everybody at the end. I'm curious to see what your responses are. So first of all, what's your favorite dairy-related book or resource?
Well, it has to be NASM 2021, right? I mean, there's no way I can answer anything else other than that.
Yep. Okay, that's the Nutrient Requirements book. So what about your favorite book or resource outside of agriculture?
I forgot who recommended it, but it might have been... it was somebody at Davis that recommended that I read The Ascent of Man by Jacob Bernowski. It was written in the 70s or 80s. It's a science book, but he was a physicist, if I remember correct. And he just talked about, the adoption of sort of hypothesis-based science, through the 13, 14, 15, up to today. I just found that a really inspiring book. I really liked that book.
Cool. I've heard of it. I've never heard anybody recommend it to me. I'll have to put that on my list. And then last of all, in your opinion, what sets successful dairy professionals apart from those who are less successful?
Well, I think obviously it's the ones that, technology keeps moving forward, the ones that continue to, try to learn new concepts and new technologies and adopt them in their day-to-day work and try to make them work for them and particularly for their farmers, are the ones that remain successful.
Well said.
I worked with a lot of different people. There was about 200 different salespeople that worked for Purina, and then there was more when Land O'Lakes got added in. And there was a huge range. There was a huge range of success as well. The ones that adopted new stuff tended to be much more successful than the ones that didn't.
Very well said. Thank you. Dr. Hanigan, thanks for being part of the show. This has been a really fun conversation for me.
My pleasure.
All right, well, this has been another episode of the Dairy Podcast Show. Don't forget to subscribe or like if you haven't followed us yet, because I promise the next episode will be a good one as well. Until then, we'll see you next time.